Hi guys, welcome back to BioBuddy and welcome back to my channel. So we are back with another video, and this video is about genomic library construction. So I will make another video about the cDNA library construction also, but I have made a video about cDNA synthesis previously. So if you want, you can watch that too. Very simple, very easy, specific terms are being used so that you people can understand it in a very simpler way. So now let's go on to the genomic library construction. Let me just take a marker. Yes, so in this process, what is happening, genomic library construction, that means in that library the entire genome of a particular organism or a species is present, right? So now it is larger as compared to the cDNA library construction. Obviously in that only the cDNA is present, but in this what is happening, the entire genome is present, right? Okay, so guys, in this we are taking the host cell DNA. We are only targeting the DNA present inside the host cell, right? So we had taken the DNA, it is taken out, and now that isolated genomic DNA, that means we are isolating the genomic DNA from the host cell. Now we are taking a vector and we are taking this genomic DNA. Two things have been taken. So now what we will do, we will digest this so that they are made capable of ligating them together so that we can insert further into the host cell. So now what we are doing, we are taking the genomic DNA. This genomic DNA is taken and digested using restriction endonuclease. Restriction endonuclease helps in the digestion process, that is, it helps in cutting it helps in cutting the DNA or cutting the digestive vector from a particular site, which helps in the process of further constructing the genomic library. Right now what we are doing, this isolated genomic DNA is broken down into many fragments. These are the given fragments, and these fragments, they are further separated on the basis of their size with the help of gel electrophoresis. As you know, gel electrophoresis, what happens, it works on the principle of plus and minus. It helps, it takes the help of the electricity, and with the help of that, the separation of the strands takes place on the basis of size. And how the process takes place, you all must be knowing, because obviously if you are studying this, you should already know what is the meaning of agarose gel electrophoresis. In that, we are using agarose gel, and on the agarose gel we run it, and when we run it, what happens, the strands are separated on the basis of their size, right? So now what is happening, this DNA fragment, they are separated by agarose gel electrophoresis. Now after this process, what is happening, ligation is also done, digestion is also done of the vector. That vector we are taking here, we are taking this vector, we also want to digest it, so digestion is also done by restriction endonuclease. Now after the digestion is done, these two have to be ligated together. These two have to be ligated together, any one of the three strands that you want to ligate. So how is it done? By DNA ligase. DNA ligase is used for bringing the two strands, bringing the strand of the DNA or the DNA fragment to join with the vector. This is done. Now obviously they will come together, they will bind together, and they will form a single fragment. Now this is formed, and this is the final result that we get. This final result is, we have to insert this rDNA into the bacteria. Now this rDNA has to be inserted into which bacteria? Mostly we are using E. coli, so we can use E. coli as the if you want to use, yes, we can use E. coli for that. After using this E. coli, we are inserting this vector into the host. Now you know what it does, it takes up the whole machinery of the cell and it uses its own, right? It synthesizes its own processes. वो अपना process करना शुरू कर देता है. So now here amplification takes place, right? We are growing them into the agar plate, and in the agar plate they further multiply, they form colonies, and that is why their amplification is being done, and further the genomic library is formed. Now what are the points which are genomic, which are in the genomic library and which are not in the cDNA library? So let me tell you that genomic library is, guys, it is larger as compared to the cDNA library, and cDNA library is only carrying the expressed gene sequence. cDNA carries only the expressed gene sequence, and it is smaller as compared to the genomic library construction. But guys, I think there are a lot of differences, so I will make a separate video for this. So right now I think you will be clear with the genomic library construction, how genomic library is being constructed. It's a very simple process, right? So let's, if you like the video, I hope you have understood, right? So if you like the video, please click the like button, subscribe to my channel for further updates so that you can get and press the bell icon, guys. So thank you.